Today we will be reading Unit 3, Lesson 2, Genetic Engineering, Exploration 1, pages 158 to 165. This cat is producing proteins that cause it to glow green under certain types of light. By the end of this lesson, you will be able to explain how genetic engineering can be used to influence traits in organisms. Can you explain it? How can goats produce spider silk proteins? Spiders make silk, a type of protein that is light, strong, and flexible. Scientists have developed goats that produce spider silk proteins in their milk. The spider silk protein can be separated from the milk and then used by humans. So question 1 asks you, what are the advantages of getting spider silk from goats instead of spiders? So here's the goat, here's the spider. The benefit of getting spider silk from goats is that goats are larger, so they'll be able to make more silk faster than the spider can. Goats are also easier to catch and easier to take care of than a little tiny spider, so that's the benefits to using a goat versus a spider. Question 2, do you think people could breed goats to produce spider silk proteins? Explain your answer. No, because goats naturally can't produce spider silk. It's a gene that scientists have inserted into the DNA of the goats. They took it from the spider and inserted it into the goats, so their milk will produce that protein. But they don't do it naturally, so you therefore can't breed a goat to get goats that do that in nature. Turn the page. Exploration 1, Exploring Genetic Engineering Techniques. You may have friends with blue, green, or brown eyes. Eye color is an inherited trait that is passed from parent to offspring. Traits are controlled by genes. Genes are segments of DNA on chromosomes. Genes code for proteins that cause specific traits in an organism, such as eye color. Scientists can change genes to influence traits in organisms. Question 3. Eye color is a trait influenced by several genes. The OCA2 and HERC2 genes determine how much melanin, which is the pigment that causes skin and eye color, is produced in iron cells. So the more melanin you have, the darker your eyes are going to appear. Same thing with your skin. The more melanin you have in your skin tone, the darker your skin tone is going to be. So people with lots of melanin have dark skin, and people with very little melanin have light skin. So like albinos, it's the reason that they are so white, so pale, is because they have little to no melanin in their skin. So less melanin would then lead to blue eyes. So complete the diagram using the terms in the word bank. So our word bank, we have proteins, DNA, traits, and genes. So starting in this picture over here, this would be your DNA. In the middle would be, so the DNA. These different parts of the DNA would be your traits. These clusters would be the proteins. These would be the genes on the DNA. This would be the proteins that the genes would make, and then these proteins would code for these specific traits. Genetic engineering is biotechnology. As scientists improve their understanding of the biological processes that connect genes to traits, they develop technologies to study and influence desired traits in organisms. Genetic engineering is the process of modifying DNA for practical purposes. Scientists might modify DNA for use in research, medicine, or agriculture. For example, soybean plants are important for oils and livestock feed. Soybeans are a genetically modified crop. Soybean crop health improved after scientists inserted genes for pest resistance into soybean seeds. Gene insertion is just one biotechnology used by genetic engineers. Other genetic engineering technologies encourage the production of specific proteins or change genes to modify traits in organisms. So a caterpillar chewed holes in the leaves of the soybean plant. Inserting a pest-resistant gene into the soybeans can decrease the damage due to pests. So agriculture is where we use genetic engineering a lot, actually. So you can see the holes from the insects. Comparing genetic engineering and artificial selection. Humans have influenced the traits of organisms for thousands of years through artificial selection. So artificial selection is the process of breeding organisms to increase the frequency of a desired trait in a population. So artificial selection is done by humans, and the trait is already there naturally in that organism, and they're just making sure that they're selecting specific organisms to breed together so their offspring will have those traits. but the trait already is there. Both artificial selection and genetic engineering influence the traits present in a population. For example, humans can improve pest resistance in soybeans by breeding plants that show increased resistance to pests, or by inserting a pest-resistant gene into a plant. Influencing traits through genetic engineering can be more precise and faster than using artificial selection. So genetic engineering is when they are tampering with the DNA. They're inserting or taking out specific genes in order to get a specific result. So it's not necessarily something that is naturally occurring in that plant. In some cases, genetic engineering allows scientists to make changes to the traits of an organism that would not be possible through artificial selection. So like the goats with the spider silk. Goats can't do that on their own, but tampering with their genetics lets them all of a sudden be able to do that. So inserting a gene into a bacterium. Bacterium is a single bacteria. Segments of DNA from one organism may be integrated into the DNA of a different organism. The simple structure of bacterial cells makes them good candidates for genetic engineering. So scientists would locate and isolate the gene that causes a desirable trait. The isolated gene is inserted into the bacterial DNA. The bacterial DNA is inserted into a bacterium, so a single bacteria. The new trait is expressed when the bacterium uses the inserted gene to make proteins. So they look at the DNA of the organism that has the trait that they want, which is that R. They take that gene out. They insert that gene into the DNA of the bacteria, and then they insert that into the DNA of bacteria, and then they insert that DNA into the bacteria to change it. So question 4, what is required to insert a new gene into a bacterium and for the bacteria to show the new trait? Select all that apply. So you have A, proteins, B, biotechnology, C, cellular processes, and D, a breeding program. Well, for all of these, all would be correct except for the breeding program. You do need proteins in order to insert a new gene. That's how genes are coded, is through proteins. You do need to use biotechnology because humans need to do it. It does require cellular processes because it requires that cell to be able to use that DNA to make whatever it's making. It might to make new proteins or to be able to do a specific function, but you do not need a breeding program. Bacteria don't really breed. They're usually it's not really asexual reproduction, kind of. So bacteria don't require sexual reproduction is basically what I'm saying. A breeding program would be for those organisms that do sexual reproduction, which means you need two parents, but a bacteria doesn't need that in order to make new bacteria. It does it by itself. So a breeding program isn't necessary. Question 5, discuss. When inserting a new gene into an organism, the gene could come from the same species or it could come from a different species. Generate at least 3 questions you could research about each of these scenarios. So you just have to come up with some questions. So things would be like, how is genetic engineering using genes from the same species to help humans? Are organisms harmed when they take the genes from the same species and insert it into the DNA? Who controls the rules that govern genetic engineering? What are the risks of inserting genes from different species into an organism? How is DNA different from a different species inserted into an organism? And then why do scientists want to create like that type of a hybrid? Are just some example questions. Turn the page. Milestones in genetic engineering. Rapid advancements in genetic engineering began in the early 1970s. Biologists saw that bacteria use special enzymes to repair their DNA. For example, bacteria use enzymes to cut and remove DNA that was inserted by a virus. Viruses are small particles that insert viral genetic information into host cells. The discovery about how bacteria could cut and paste enzymes inspired scientists to investigate how these enzymes could be used to solve human problems. Developments in genetic engineering reflect scientific scientific progress. Scientists use creativity and existing technology to make new discoveries. This leads to increased understanding and chances for more exploration. So viruses are living. Viruses, kind of think of them kind of like little tiny microscopic robots. It's kind of how they kind of work. So these robots are programmed to find living things, living cells, and they latch onto those cells and inside the virus is genetic material. So that genetic material is then injected or put inside that living cell, and that genetic material tampers and makes that cell a bad cell, a rogue cell. And that rogue cell will then infect other cells. So that's how a virus works. It's not living, but it has genetic material in it, and that's what it uses to infect cells. So recombination technology. When a new gene is inserted into DNA, the DNA is called recombinant DNA. Recombinant DNA began by using plasmids, or small circles of bacterial DNA. So that was what the picture I showed you before with the circle that they put the R into. So step 1, the gene that controls a trait is located and cut from the chromosome. Step 2, the plasmid is opened using cut enzymes. Step 3, the new gene is added to the plasmid using paste enzymes. Step 4, the plasmid with the new gene is inserted into the bacterium. And step 5, the bacterium produces the new protein. So step 1, they locate the gene that they want, and they use the cut, and cut it from they take it out, cut it from the chromosome, so that R. Then step 2, the plasmid, this ring, they use a cut enzyme to make an incision into that enzyme, or into the plasmid. They have a cut enzyme to make an incision into the plasmid. Then they use a paste enzyme to put that R that they just took out from this strand, to put it into that plasmid. Then they inject that plasmid into the new gene, with the new gene, into the bacteria, and that bacteria will make a new protein. So question 6, number the milestones of genetic engineering to show the progression of science. So we have foreign DNA successfully inserted into the DNA of bacteria. Which would be, well, I'll read them all. Scientists confirm that the new gene gives the bacteria new traits. Cut and paste enzymes are discovered by observing new bacteria use enzymes to destroy the DNA of invading viruses, and bacteria use inserted genes to make proteins, just as they do with their own genes. So from top to bottom, they would be 2, 4, 1, 3. So first would be cut and paste enzymes are discovered by observing how bacteria use enzymes to destroy the DNA of invading viruses. That leads to foreign DNA is successfully inserted into the DNA of bacteria, which led to bacteria use inserted genes to make proteins, just as they do with their own genes, which led to scientists confirming that the new genes give the bacteria new traits. We are skipping page 163 because it's allowed, and we're skipping the top of page 164. Influencing traits in a population. Engineers can insert genes that cause desirable traits in an organism. In order to be heritable, the modification must be made to cells passed to offspring. This is easy in bacteria. Bacteria divide in half and pass a complete copy of their DNA to their offspring. In sexually reproducing organisms, genetic modifications will only be heritable if it occurs in the reproductive cells, eggs or sperm, or in early embryos. This makes it more difficult to use genetic engineering to influence traits in a population of organisms that reproduce sexually compared to bacteria. The cells in early embryos can still develop into any cell type. They pass an identical copy of their DNA to every offspring cell, including any genetic modifications. During the process of cell differentiation, cells become specialized for a specific function. Once cells differentiate into various types, such as muscle cells or blood cells, genetic modifications made to the cells will only pass to the offspring cells of that cell type. So cell differentiation in embryos. Early embryo cells differentiate into different cell types found in an organism. This diagram shows the 3 examples of specialized cells. So in humans, we have different types of cells. So like we have nerve cells in our brain, we have muscle cells, and we have blood cells. So the cells of the early embryo would evolve into those 3 cell types. So that's cell differentiation. So the embryo cells turn into nerve cells, muscle cells, and blood cells. Page 165. Did I skip a question? No, I did. Okay. Do the math. If all the poisonous frog eggs are genetically engineered right after fertilization to include a gene that prevents poison production, what is the ratio of poisonous to non-poisonous frogs in the offspring? So you have A, 1 to 0, B, 1 to 2, C, 0 to 1, or D, 1 to 1. So basically they put in the embryos, or they made the eggs They included in the eggs a gene that prevents poison production. So what is the ratio of poisonous to non-poisonous frogs? Well, the answer should be C. You should get no poisonous frogs. All the offspring should be non-poisonous. So you should get no poisonous frogs, and all your offspring should be non-poisonous, because they have the gene that prevents the toxic production because it was inserted into the egg cell. We're skipping question 8. Genetic modification of bacteria. Genetic engineering can be used to help solve human problems. People with diabetes need a dependable supply of insulin. Insulin is a protein that helps regulate blood sugar. Scientists have modified bacteria to produce insulin for human use. Large numbers of modified bacteria can be farmed to produce large amounts of insulin. So genetic modification of bacteria to produce insulin. So they isolate the insulin gene from the human cell. They cut the plasmid DNA in the bacteria. They insert that insulin gene into the plasmid DNA, and then they insert that plasmid DNA into bacteria to produce insulin. So the genetically engineered plasmid spreads through a population of bacteria, and it will be passed from the parent to the offspring. So question 9, why is it necessary to genetically engineer the bacteria to produce insulin instead of inserting the insulin gene into humans? Scientists modify bacteria because it's easier to modify a bacteria than it is to modify the human. The human reproduces sexually. The bacteria reproduces, we'll classify it as asexually. So bacteria don't require two parents. There's less genes that because everything gets copied and passed to the offspring. You're not getting different combinations of genes because it's an exact copy of the parent. So it's easier to change and add things into the bacteria and get the results you want than it is to change and add things into the human and try to get that insulin production. Your assignment today is a gene therapy GMO worksheet. It's 10 questions. For the gene therapy, you pick a disease, research about it, and then answer the questions about it. And then for GMO, same thing. You're just going to look up some information about genetically modified organisms.